I am Magdalena Sevilla, and I am postdoctoral fellow with Dr. Elisa Manning at Massachusetts General Hospital. Next slide, please. So I'm happy to share the project that I've been working with BioData Catalyst with all of you. My project entitles a comprehensive analysis of omic signatures across ethnic groups and its implications for cardiometabolic diseases. The advent of omics studies over the last decade offers an important opportunity to understand the processes involved in the physiopathology of complex diseases such as type 2 diabetes. We all know that there are considerable differences in the prevalence of type 2 diabetes and other diseases, as we just saw, across race, ethnicity. And at least for genetic studies, the transethnic approaches have increased the understanding of the physiopathology, particularly for type 2 diabetes, by improving power and increasing resolution for fine mapping. However, most omics studies, particularly for omics, most of them have been conducted in European individuals, and the extent to which this results can be translatable to other populations with different environmental exposures and also different allelic frequencies is already unknown. Next slide, please. So the study that I am proposing for BioData Catalyst was to identify the omic specific signatures or the variation in the profiles of different omic types across different ethnic groups. And the top med studies or cohorts that are implicated in this project are the multi-ethnic study of atherosclerosis, MESA, and Women's Health Initiative. So we have considered 4 different data types, omic types: metabolomics, proteomics, transcriptomics, and epigenomics. And we have applied 5 different steps for each one of these omic types in each one of the cohorts: data acquisition, data harmonization, data cleaning, and association analysis. We have performed QC for each one of the data types, considering the particularities of each data type. And we have performed an association analysis using LIMA, a bioconductor package that performs linear models using Bayesian methods for more stable results, considering that these data types tend to be high dimensional, and also they showed a high correlation among each one of the signatures. Next slide, please. Thank you. So after applying our association analysis, we ended up with a set of significant signatures. It could be, this is the example of the metabolomic signatures in MESA only, but we have identified differential signatures for each one of the ethnic groups in MESA and also WHI. We have contrasted each one of the ethnic groups versus the rest of the population. And this allows us to identify the distinctive and also the common signatures across the different ethnic groups. So we are in the process to understand how these differences can affect potential cardiometabolic outcomes by trying to understand the role that that each one of the signatures play into their physiological pathways. Next slide, please. So in case of gene expression and also metabolomics and proteomics, we have run in pathway analysis, and we have identified some possible signatures that can be affected in a different way by ethnic group. For example, in the sphingolipid signaling pathway, we observed a transcript, ADORA1, that has a potential distinctive role between each in different ethnic groups. For example, it has a positive association in African American individuals versus the rest of the sample, contrasting with a negative association in white individuals versus the rest of the sample. However, these results are very preliminary and we're still working on them. Next slide, please. Thank you. So what components of the ecosystem has been helpful for me? I would say that BioData Catalyst has been an incredible resource and tool that has allowed me to have a repository because it has been very helpful to have a single place to storage all the data types and also across these two cohorts. So we have used as a data storage and also data sharing with collaborators as well. So this has been an incredible tool for this. Also, I have used, for example, WIDLs in this case to download dbGaP data from dbGaP. This WIDL was created by a former fellow, Kenny Westerman, and it has been very helpful to download all the data types that we needed from all the cohorts and to keep them in the same format within a same workspace, which is very nice. And then, well, I've been working with notebooks for the association analysis, principal component analysis, pathway analysis, and this allows the reproducibility of the analysis across the different cohorts and also in some cases across the different data types. So also the fact that we can share this with other analysts in the team has been incredibly helpful. Finally, I am becoming more familiar with Picture. Picture is a tool that allows us to identify the variables that each cohort has or that are available for us in a friendly environment to select them. So I am becoming more familiar with this tool, and I think that since we'll be incorporating environmental variables into our analysis, this will be a very interesting tool to apply. So in summary, I've been using WIDLs, notebooks for data storage, sharing, download data from dbGaP to perform association analysis from gene expression, metabolomics, and proteomics so far. And we are working in creating pipelines to integrate different omic types using weighted gene correlation network analysis or WGCNA, and also sparse canonical correlation analysis as well. And that is in progress. Next slide, please. So I would like to highlight the things that really helped me to accomplish my research or that that has been very helpful even now. I don't come from a computational background. I have a clinical background. So this tool has been incredible help. It has been very helpful for me to have a smooth transition because at least from the Terra platform, we have available a lot of resources, documentation, tutorials, blogs is frequently asked questions. And I think it's very, very helpful to have all these resources in hand. Also, the Terra support team has been incredibly helpful. They are very responsive, and even though if the first person that you approach doesn't have the answer, they will help you to find the person that can help you with your problem. Other thing, I would say that having other lab mates and collaborators that use the same ecosystem has been very helpful because we can share the data very easily and also share WIDLs and this other tools. So I think that is also very helpful. And finally, the BioData Catalyst community, the community of fellows, the working groups, I think this is also a very nice resource. Next slide, please. Finally, some advices. Well, I think that identify key resources and also don't be afraid to ask because we come from different backgrounds, so it doesn't matter the question. I think it's always helpful for everyone. So I think that's important. And also something that it's important to identify other collaborators or other fellows that have worked with similar data types or also similar analysis, so we can share resources or whatever. So I think that share is something important, either experiences, resources with other fellows that also help others and help you to identify potential problems. But yeah, basically I would say that share is the most important part of this. So thank you very much. Happy to take questions if there is time.
I think we have time for 1 or 2 questions before we go on to the next speaker. You can either ask them in the chat or if you want to unmute yourself and ask out loud.
Hi, Magda. This is Yelling. I'm curious that the metabolomics that you have found, is this stratified by population with type 2 diabetes or not?
No, we haven't considered, the only covariates that we have considered so far are sex and age. So the next step of our analysis is to create a mediation type of analysis to understand whether the signatures can affect type 2 diabetes prevalence, for example. But that is the next step of our analysis.
But these signals are all associated with the CVDs in MESA, right? That you found.
Yeah, well, or
yeah.
Or variable,
No.
Well, there's no association with CVD now. We only have the association with ancestry, by ancestry.
Okay.
So far, that's our only variable that we are considering, adjusting by sex and age. But we haven't performed any association with any other trait yet.